Okay. So good morning, everyone, and welcome to our regular PAASE webinars. So our speaker for today is the Vice President and Head of Global Research and External Innovation at Sanofi Pasteur. He is responsible for vaccine research and search and evaluation function for external vaccine and vaccine technology opportunities. He has more than 2 decades of experience in research and development of vaccines and biologics in both pharmaceutical and non-profit sectors. Prior to joining Sanofi Pasteur on November 2017, he was the Chief Scientific Officer at ARAS, a nonprofit organization dedicated to the development of new and more effective tuberculosis vaccines. He played a leading role in developing the long-term scientific strategy for the field and advancing promising candidates to the development stages. During his 19 years at the Merck Research Laboratories, he contributed to the licensure of Merck's papillomavirus vaccines and the development of novel vaccine candidates against several viral pathogens, including herpes viruses, Ebola, HIV-1, RSV, and dengue, and monoclonal antibodies against respiratory pathogens and oncology targets. As a prominent scientific leader, he has also served on numerous advisory committees to the Bill and Melinda Gates Foundation Global Health Program, International AIDS Vaccine Initiative, NIH Integrated Preclinical Clinical AIDS Vaccine Development Program, U.S. Agency for International Development, HIV Vaccine Enterprise, and Human Vaccine Project. He received his bachelor's and PhD degrees in chemistry from the University of the Philippines and the California Institute of Technology, respectively. Let us all welcome our speaker for today, Dr. Danilo Casimiro, to tell us more about the race to addressing the global COVID-19 pandemic through vaccination. So thank you. Take it away, Danny.
Yeah, can you hear me very
Yes, we
well?
can hear you.
Okay. Thank you, Kathleen, for the kind introduction, and thank you very much for the Academy for the invitation to speak to you today. So I'll bypass this slide because this Kathleen had gone through at least my background coming into to the talk today. So let's get to the subject at hand. Um, the Everyone's very familiar with these numbers. The COVID-19 pandemic is probably one of the most, if not the most significant public health emergencies in the last 2 decades. There are 61 million cases to date, 1.4 million deaths, and over 200 countries reporting infections with the COVID-2 virus. Uh, the, the graph you see here are the number of cases as a function of time in different regions of the world, the Americas, Europe, Southeast Asia, Eastern Mediterranean, Africa, and Western Pacific. The COVID-19 disease is caused by what is termed as a SARS-CoV2. Remember, about a few years ago, there was SARS-CoV that was designated now as COVID or COVID-1. This is now COVID-2. Now, the coronavirus phylogenetic is shown right here. There are four major families: alpha, beta, gamma, and delta. And the pandemic strains arise from the beta family of coronaviruses. I show in here too as well some of the human coronaviruses. Coronaviruses are really prevalent, right? I mean, for human pathology. They cause your common cold, along with a number of other pathogens. But the more severe ones are the pandemic strains, the MERS, SARS, and COVID-2. Now, MERS came up in 2012, SARS-CoV or COVID-1 in 2002, and COVID-2 in 2019. And remember, there was also an Ebola outbreak in 2013. So just in the last decade, there have been a high increasing frequency of pandemics in the world, starting with MERS, Ebola, and now COVID-2. The genome structure is very simple, right? These are the number of genes, open reading frames for COVID, and they correspond to a number of structural proteins on the virus itself. The most prominent one is what we call the spike protein. This is the one that really protrudes out of the virus. And one additional note is that Since the emergence of COVID-2, there have been alterations or mutations in the virus. The most prominent one is the emergence of the aspartic acid 614 to glycine mutation. This increased the infectivity of the virus as well as the transmissibility of the virus. And typically in humans, they amount to higher titers or infectious or viral loads in the individual. So the current pandemic is now dominated by this mutant of the original COVID-2. This slide shows you the disease associated with COVID. Now, in terms of severity, there's a great majority of people, this is the frequency, are asymptomatic. But as you go from asymptomatic to mild, moderate to severe disease, initially fever, cough, diarrhea, loss of smell, loss of taste, shortness of breath, and that could also progress to acute respiratory distress, wherein fluids are going in now inside the lungs. The frequency obviously is highest with asymptomatic and goes down with the severe disease. But what is constant, whether you're asymptomatic or have severe disease, is that you are shedding virus up to 14 days. And that's why it's important to observe the social distancing and mask wearing, because asymptomatics are carriers for the virus itself. We all know about the high mortality rates often associated with age, over 70 years of age, or a number of comorbidities, cardiovascular disease, diabetes, obesity, chronic kidney disease, COPD. Now, in order to develop vaccines, we need to understand what the virus does. But before going there, there are three ways, or in some cases more than three ways, to tackle the infection and disease. One is through chemical drugs, and these are typically used to treat disease symptoms or for therapeutic purposes. Antibodies are also in development for COVID-2, primarily for treatment or post-exposure prophylaxis, meaning if you suspect you were infected, you have some mild symptoms, you can get on prophylaxis with antibodies to prevent the progression to severe disease. And then we all know about vaccines. Vaccines are there to prevent infection, and in most cases, let me just stress, vaccines are primarily to prevent disease. If you look at all the labels, all the vaccine, their labels are prevention of disease, not prevention of infection, right? And if you look at the public health impact of vaccines to control global infectious disease, this is one figure, very powerful figure from Forbes magazine, where in the US, the pre-vaccine era, these are the number of cases per year for diphtheria, Haemophilus influenza, hepatitis A, hepatitis B, measles, all the way down, rubella, smallpox, pneumococcal diseases. With the introduction of vaccines, you see a 91 to 100% reduction in disease burden caused by a number of these pathogens. Right? So this tells you how really powerful these vaccines are. It changed the whole world. Let me just say, no one has won yet the Nobel Prize for vaccines, but one has to look at the possibility of, at some point, a Nobel Prize winner for vaccines, because you can see the public health impact of vaccines here. And these are just for Sanofi. These are the pathogens for which we have products in the US. What should a COVID-19 vaccine do? As I mentioned, vaccines are really meant to prevent disease, right? So disease is always, the disease prevention is always the primary endpoint in a clinical trial, a licensure trial for a vaccine. It's there to reduce morbidity as well as mortality rates and reduce hospitalizations, right? Now, one would hope that if you have a vaccine, a fairly potent vaccine, you could also prevent acquisition or infection by an individual. Now, the implications of that one is that you will reduce transmission, especially As we know, many of the infections lead to asymptomatic cases. And that's really important to establish herd immunity. This is the primary endpoint for all the vaccines in development today. And there are vaccines where they are looking at as secondary endpoint the prevention of acquisition. Now, let me just go into what makes up a vaccine, right? There are many components of a vaccine, but I want to highlight two. One is the antigen. That the antigen is the component of the pathogen, right? In this case, COVID-2, to which an immune response is induced against. And that immune response is intended to provide protection against the infection or disease. So one has to understand what is the important pathogen in the important component, a protein or a polysaccharide or a glycosylated protein that would serve as the component of the vaccine to which you want to induce the protective immunity against. The other component or consideration is how would you deliver the antigen to the individual? There are numerous technologies which are in play in terms of delivering antigens to the human subject. And we hear about, I'll explain later on about mRNA, DNA, viral vector, and protein adjuvants or adjuvanted proteins. Now, to understand what would be the most suitable or appropriate target for a vaccine response, one has to understand the life cycle of SARS-CoV-2. Now, the virus itself will attach to a receptor. This receptor is known as angiotensin converting enzyme 2, which is uh, widely expressed in numerous tissues. Uh, the virus itself would attach to the receptor. There's a number of events that will lead to fusion of the virus with the host membrane. That fusion will release the genome. The genome itself will produce enzymes which will be responsible for further replication or production of progeny of additional genomes. Those genomes will then express a number of proteins which will now be inside the progeny virus. And then the virus itself is released from the cell and you have progeny virus itself, right? So in order to have an effective immune response, the most obvious place typically for virus is to block the attachment to the receptor itself. And that can be done by inducing antibodies that would recognize or bind tightly to the relevant antigen on the surface of the virus, precluding the ability of the virus to attach itself to the host receptor. If you look at the structure of COVID-2, that protein which is responsible for attachment is called the S or the spike protein. That's mainly the protrusion on the virus itself, on the surface of the virus. This is the structure or the primary structure of the spike protein expressed by the virus. There are two components, the S1 and the S2. They are cleaved upon attachment on the host cells through what is known as a furin cleavage site. Furin is a host protease. It's found on many human cells. So it's actually using a human protease to cleave the S protein to S1, S2, and promote infection of the virus. And that's done this way, right? So here's the virus. Here's the host cell with the ACE2 receptor, and here's the virus with the protrusion, which is the spike protein, S1 and S2. Upon attachment of or upon binding of the S protein to the receptor, the furin will cleave S1 from S2. So now you'll have S1 bound to the receptor. The S2 itself will undergo a conformational change such that it inserts itself onto the host cell membrane. A second conformational, this is called as a pre-hairpin structure. And a second conformational change will force these three helices to form what is known as a six helix bundle. The six helix bundle will bring now the virus membrane with the host cell membrane, promoting fusion. In order to block this event, one way of doing that is just merely blocking the attachment of the S protein to the angiotensin, to the ACE2 receptor. Or further down in the process, you could also block the formation of the six helix bundle. But we're going to focus on the inhibition of binding of the S protein for the virus to the angiotensin converting enzyme 2 receptor. Now, when it comes to developing a vaccine against a pandemic, there is the element of time. It's a pandemic, right? This is a typical course of developing a vaccine. Research, manufacturing the vaccine for phase 1, some toxicology studies, and then the phase 1 trial, which is a safety immunogenicity study, and then manufacturing again for the materials for the phase 2 study, which will typically are dose refinement studies, and then manufacturing industrialization efforts before you start the large phase 3 study, which will be the field efficacy study for the vaccine. There's the biologics BLA submission to the agency and launch of the vaccine. Typically, this would take more than 10 years, but we don't have 10 years for COVID-19, right? So what are the considerations when it comes to developing an R&D program for a COVID-19 vaccine? One is speed to clinic and speed to licensure. So I mentioned this does not apply, cannot be applied for a pandemic vaccine development. You have to be months from laboratory to the phase 1 trial, and months, if not no more than a year, from phase 1 to licensure. This really favors a number of the novel approaches, and I'll explain why later on, and not the traditional approaches for vaccines. The other consideration is that you have to have a large-scale manufacturing capacity at hand and ready to go, right? It's one thing to license a vaccine, but if you can't deliver the number of doses to address the global pandemic, then you're screwed in itself. Typically, you have to start delivering supplying the global and regional needs within months, if not no more than a year, right? After licensure of the vaccine. Now, this favors traditional approaches because with traditional approaches for vaccines, the manufacturing infrastructure already exists for large-scale supply manufacturing. If you're dealing with a novel approach, meaning there's no vaccine really licensed using that particular platform, then you would require more industrialization development. And by traditional and novel, I mean the following. So I've explained now how we chose the antigen. The next question is, how do we deliver the antigen? The traditional approaches are, well, it's an antigen, it's a protein vaccine. Why don't we just make the antigen recombinantly, right? So protein subunit vaccines. And there are vaccines using this platform against hepatitis B, meninge B, the papillomavirus, which is now a 9-valent vaccine. It's a recombinant protein expressed in cell culture. Inactivated virus vaccines, if there is a way of delivering the whole virus safely through inactivation, then you can do so. And the number of vaccines, inactivated virus vaccines exist for hepatitis A, influenza, and rabies. Now, the more novel technologies for which there are no licensed vaccines are DNA vaccines, mRNA vaccines, and viral vector vaccines. This is a summary, at least about a couple of months ago, I'm not quite sure if it's updated yet, of the number of vaccines in the preclinical and clinical development against COVID-19, right? Majority of them are, this is a global outlook, majority of them are protein-based vaccines. a number of viral vaccines, and then mRNA vaccines, DNA vaccines, and all the other flavor or technologies for vaccine R&D. I'll explain a bit about those novel approaches and compare it to the traditional one. The traditional one, so we now know that the S protein is the antigen that we need to generate protective immunity against, right? So if you have the DNA itself, you can, for a subunit vaccine, you can use the DNA you can transfer the DNA through transfection or infection with, say, a baculovirus into mammalian cells. You culture the cells in large fermenters, isolate, and then purify the protein. This is the protein itself. This is the S protein. And in some cases, my apologies there, if the protein is not immunogenic enough, we do add an adjuvant. In the case of COVID-19, the adjuvants that is most commonly used are what you see here. These are oil in water emulsions, very small microparticles of oil in water. This is the vaccine. This vaccine right here, protein plus adjuvant, is the one Sanofi is working with GSK. Novavax is another company in clinical trials with an adjuvanted protein. Clover Pharma also has an adjuvanted protein. Now let's go next to what an mRNA vaccine is. The mRNA vaccine is that you start with the DNA, you transcribe the DNA in vitro using cell-free manufacturing into mRNA. You purify the mRNA and then you package that into a synthetic lipid nanoparticle, which is able to infect and deliver the mRNA into the host cell. So this primarily is the vaccine. You're not delivering the protein, you're not giving the protein itself, but you're actually delivering the coding sequence, the mRNA that codes for the protein. So you're using your muscle cells to manufacture the antigen. So one can look at this one as a synthetic virus, right? This is the vaccine platform for Pfizer, BioNTech, Moderna, CureVac, and Arcturus. I'm highlighting here the vaccine companies in clinical trials today. The last one is the viral vector. If you can take the DNA itself, insert that or clone that into a live attenuated adenovirus. In the case of what's in the clinic right now, there are adenoviruses. Adenoviruses cause the common cold, right? But they are further attenuated, but they are very efficient, being they are virus. They are very efficient in delivering the DNA inside the cell. You now grow the virus in cell culture, isolate the virus, and that's the vaccine itself. So you're using the virus to deliver the coding sequence into the host cell, and the host cell will manufacture the antigen for you. So these two are what we call genetic immunization, and this is the recombinant protein. Very traditional way of delivering vaccine antigens, protein with adjuvant. I'll have only one slide for Sanofi Pasteur's approach. We have two ongoing programs right now. One is in the clinic. The first one is a recombinant subunit vaccine approach or vaccine candidate, which is the protein antigen produced in insect cells and then combined with an oil in water emulsion in collaboration with GSK. The advantages are we do have existing large-scale capacity for manufacturing. Should this be licensed, Sanofi can supply up to 1 billion doses by within 1 year of licensure. The phase 1/2 study started in September, and if everything goes well, we anticipate earliest potential approval in mid-2021. The second approach is the mRNA vaccine in collaboration with a company called Translate Bio. We have a broad infectious disease vaccine license with Translate Bio. Again, it's an innovative approach. It has significant existing investment in mRNA capacity because Translate Bio was part of a pharmaceutical company. So they really invested in large-scale manufacturing of the mRNA vaccine. At the phase 1/2 study start, we anticipate that one in mid 1Q 2021. And we anticipate also a capacity of 90 to 360 million doses annually, depending on the vaccine dose that will be licensed. Let's now go to an example of an mRNA vaccine. So I don't want to highlight all the vaccines, but let's just look at Moderna's mRNA vaccine because that has really generated a lot of interest and a lot of publicity in the last few months. I'll show you here the efficacy of the Moderna vaccine in preclinical animal model systems. It's been shown to be immunogenic and efficacious against SARS-CoV-2 challenge of monkeys. So here's the study that this was published earlier this year. Uh, the, the scientists at Moderna at the NIH immunized monkeys at day 0 and day 28 with the mRNA vaccine, and then challenge those monkeys with a live COVID-2 virus given intranasally as well as intratracheally. And then they sample the bowels as well as the nasal washes for virus. At day 56, they also took blood so they can assess the immunogenicity of the vaccine. This is the neutralizing titers. This is really an in vitro assay measuring the ability of serum from vaccinated animals to neutralize the virus in culture, in a dish. Here's PBS. So we anticipate no neutralization. This is on a log scale. Here's 10 micrograms of the mRNA and 100 micrograms of the mRNA. There's a clear dose response going from 10 to 100 micrograms given 2 doses twice 4 weeks apart. Here are the titers in human samples, convalescent human samples. These are individuals who recovered from COVID-2 infection and disease. As you can see, the immune response in the monkeys are on the upper end or the upper range of human convalescent serum. If you take now the bronchoalveolar lavage samples, which is really a representation of what you can expect in the lungs, and then the nasal wash, you also see an effect of the vaccine, of the immunity to the vaccine on the viral copies or the viral loads on those 2 tissues. Here's PBS, and this is what you would expect. You see 5 to 6 log virus in the bowels in PBS at day 2, day 4, and day 7. But for the vaccinated animals, you see a clear attenuation of the number of viruses that had been isolated and measured in the bowels at day 2, 4, and 7, also at 100 microgram. This is what you see in the nasal washes. A lot more variability in terms of the viral copies in the placebo, but clearly if you compare that with 100 microgram, you see a big reduction in the viral copies in the nose in animals that were vaccinated with 100 micrograms of mRNA. Here's the phase 1 human clinical trial for Moderna 04 vaccination, 3 dose levels. They did 25, 100, 250 microgram of the mRNA. And clearly, there doesn't appear to be a difference between 100 and 250 micrograms. There is some increased reactogenicity, some trend towards increased reactogenicity at 250 microgram. So they opted for the phase 3 trial using 100 micrograms of the mRNA. So I want to give you just a picture here of how fast Moderna really develop this vaccine. It took them only 2 months from when they got the sequence from the NIH to go and start the phase one trial. Remember, in my old time frame is about 3 years, right? Conventionally for many vaccines. 2 months from here to phase one, another 2 months from phase one to starting phase two, and another 2 months starting phase two to starting phase two to starting the phase three trial. They are they're in the midst of their phase three trial, and in November 16, if you recall, they announced the interim results of their phase three trial showing a 94% efficacy of the vaccine within the first 2 weeks after the second vaccination. Right. The only data they have to share is the first 2 weeks after the second vaccination. They've been observing and soliciting cases during those first 2 weeks. And if you compare the placebo from the vaccinated cohort, there is a 94.5% reduction in the number of cases. Pfizer BioNTech also reported a 90% efficacy of their vaccine in the first week after the second vaccination. And more recently, AstraZeneca reported 70% in one of the cohort in terms of vaccine efficacy and 90% in a second cohort. The difference between the cohort is that for the 70%, they gave the vaccine at the highest dose for the first and second dose or second immunization. For the more efficacious vaccine, they gave half a human dose first time and a full human dose the second time. So the question that we all have is that how long will the vaccine-induced immunity last, right? Will this be a vaccine that we'll have to take on an annual basis, or is this a vaccine that will take every 3 years or every 5 years? We don't know the answer to that right now because the frequency of the booster is something you determine empirically from phase three as well as surveillance studies. To give you an example, I was involved in the development of Gardasil. Gardasil, when I left the company, had maintained the efficacy for as long as 9 years post the initial series of immunization. I haven't followed it since then, but it definitely provided a very durable immunity against human cervical cancer caused by human papillomavirus. Um, so let's take a look at this picture right here, right? With vaccination, as I mentioned, neutralizing antibody, the levels of neutralizing antibody correlates or is believed to correlate with protection. So with vaccination, you anticipate, and we've seen in animals as well as humans, an increase in neutralizing activity. That will decay with time. The reason for it is the following. You have, once the B cells, which are responsible for generating those antibodies, once they've been activated, many of them will develop into plasma cells. These plasma B cells are the one producing the antibodies that really circulate in the serum. Now, a fraction of these activated B cells will evolve into what we call as memory B cells, which plasma cells have weeks in terms of half-life, memory B cells 1 to 2 years, right? So you do anticipate, because plasma cells have a short half-life, this antibody level will go down. Let's imagine a case where there's a minimum threshold for sterilizing immunity, meaning immunity that will completely block virus infection. Right? If you for some reason encounter a virus, when your antibody are way above this minimum threshold, you anticipate very little infection. And in most cases, you won't even see a boosting of this response, all right? Because the virus is completely sequestered and not even presented to memory B cells for expansion. So in this case, if you have immunity above sterilizing immunity, you don't see much infection. The viral load is very minute. And obviously with very little infection, no disease. Now, at some point, the antibody levels would go below that level of sterilizing immunity. Now, does that mean you're not protected? Such is not the case, because if you have very effective memory B cells, once it encounters the virus, those memory B cells will expand immediately will develop immediately to plasma B cells, and you would see this rapid rise in antibodies, which is pretty much catching up with the virus infection, right? So if you're an individual who's not vaccinated and you see the virus for the very first time, you would see very high rise in viral load. And in most cases, if you have comorbidities or if you're an older adult, it could lead to severe disease. Now, however, if you have memory B cells that they can expand and control the virus to a level which is lower than that for a non-vaccinated individual, you do anticipate that you would now reduce the severity of disease or even the severity of the symptoms against COVID-19. Now, with this expansion, oftentimes the level that you will see of antibodies you will see would exceed what you had with the vaccination, right? Because you have memory B cells here. You didn't have memory B cells at the start of the vaccination. So you can use actually natural exposure as a means of boosting the immunity with time. So it's very likely that you may not require annual boosters with a COVID vaccine, but that remains to be seen and that remains to be determined in clinical trials. One concept I really like to leave you here is that most vaccines do not really rely on maintaining very high levels of antibodies against the virus. Most vaccines work because of this, what we call the fast boosting, the anamnestic response. Vaccines work largely because of the natural boosting, this fast, rapid rise in antibodies due to the existence of memory B cells that could expand to plasma cells and produce high levels of antibodies that would control the infection. So in summary, the global COVID-19 pandemic is caused by what Tony Fauci refers to as his nightmare pathogen, highly contagious airborne pathogen with high mortality. Right now, it's about 3%. Obviously, it's higher in older adults or individuals with comorbidities versus 1.1% for flu. COVID vaccines are showing promise in late-stage trials to potentially be the most effective tools to control the global pandemic. Let's still recognize that it is only one of many tools. Unless we have very high penetration or implementation of the vaccination, we will still have, in some cases, have to practice social distancing as well as wearing masks, right? The reason for it, just imagine if you had a vaccine that would only be rolled out or be accepted by 20-30% of individuals. If people start not wearing masks, the benefit of the vaccine will now be outweighed by people not wearing masks. So you may not see on a population level the impact of vaccines. Vaccines are being developed at pandemic pace, as I've shown you with Moderna, while still ensuring public safety, right? It's very important that in these trials, they have enough, the cohort size are large enough to ensure that the vaccine is safe and efficacious. There will be long-term follow-up of the trials, as well as post-licensure surveillance, as is necessary for all vaccine development to determine durability of protection as well as vaccine safety. Now, what I did not get into is that how is it possible that Moderna and many companies can move very quickly? One of the important contribution that enable this fast track of vaccines are really the private public partnerships. NIH, for example, the Operation Warp Speed, BARDA funding and the risking not only the development of the vaccine, phase 3 trials cost millions, hundreds of millions of dollars, right? Typically, we go to phase 3 and spend that money. Industry would spend that money only if you have some assurance the vaccine, some confidence the vaccine will work. And that's why we have a very normal, slow pace of development. But with the uh, with the inclusion of funding from BARDA, from governments, we can now de-risk vaccine development, where we can now share the risk, especially the financial costs of vaccine development, with those partners. There are also advanced market commitments, meaning governments making advanced purchases for vaccines. You've seen that for AstraZeneca, Sanofi, Pfizer, announcements of governments making advanced purchases for vaccines. And this is really what makes this enterprise very effective at this stage in terms of developing vaccines at such a pandemic or rapid pace. So with that, I'd like to thank you for your attention, Mike, for introduction to the academy and the folks in on the call today, and I would be happy to take some questions.
Thank you very much for that talk, Danny. I think you packed a lot of information